as far as a little history is concerned, very early on in history, people did notice that the offspring seemed to have some type of a blend of what the two parents had. So this was what was called the blending hypothesis. So they noticed, for instance, if they had a tall parent and a short parent, the offspring were medium in size. The way it's represented on this slide is that if you have a blue liquid and a yellow liquid, they're going to combine together to create a green liquid. Now that green liquid, you would not be able to get the yellow and the blue back out from it. So if that green individual was to produce offspring, it would have green to pass on to those offspring. Gregor Mendel was the one that basically disproved that blending hypothesis. So instead, he came up with what he called a particulate mechanism of inheritance. The difference here when we talk about a particulate mechanism or a particulate hypothesis is that no longer are we looking at these two parents as having something like a liquid that's going to combine together to create a new color in the offspring, but instead they have particles, or you can think of them as beads. So one parent has yellow beads, the other parent has blue beads, and those beads will combine in the offspring. When you look at that child may appear to be green, but you could still separate back out the yellow and the blue beads to pass on those separate ones to the offspring. So this is the particulate mechanism. This is what we'll be talking about in this chapter. And Gregor Mendel, he was a monk in the 1400s. So when he did his work, he was really just basing it all on numbers. He did not know what chromosomes were or DNA was at this particular moment in time. He did all of his work with the common garden pea. He worked with peas for a number of reasons, but one of the reasons was that they had a very short lifespan. So in about a 6-week time frame, he could plant the pea and have it flower and then get seeds from that particular pea plant. He also worked with them because he had a lot of varieties available to him. He did use the terms character and trait quite a lot. These are not necessarily terms that we use today because we do now know what the molecules of inheritance are. But what he called a character would be what we would call a heritable feature. And then there were traits, which were variations of that. If we look at it in things that we see today, the heritable feature would be something like eye color. And then you can have different versions of eye color, blue, green, brown, etc. He was looking at pea plants. So he was looking at seed characteristics very often. Seed shape was one of those characteristics that he looked at. So they could be round or they could be wrinkled. In all cases, when he chose what he was going to look at, he always chose things that he considered either or characters. And what that means is that there were only two possible outcomes that he could get. So when he looked at the seed color, for instance, they were either yellow or they were green. There was no in between. There were no other colors. The flower color we'll look at quite a lot. They were white or they were purple. He did have a number of traits that he looked at or characters that he looked at, and every single one of them there were just two possibilities. Another very important thing that he did was he always started with true breeding plants. By true breeding, we're talking about plants that always produce offspring like themselves when they are self-fertilized. Since we are talking about plants, we can have self-fertilization, where you can have the same plant be both the mother and the father of the offspring that are produced. And true breeding would be what we would equate to purebred. When we talk about animals that are purebred, if we breed those animals with like animals, we expect the offspring to look very similar to what the parents look like. So always he started with two true breeding parents, and those were purebred parents, essentially. In all of his work, he came up with two fundamental principles of heredity. He had the law of segregation, and then also the law of independent assortment. We're going to talk about the experiments that he did to come up with these two different laws, and then the foundation of these two laws as far as what molecules are involved and why it all works. His technique was that he would start with the two true breeding parents, and they were always contrasting parents. So we will be looking quite a lot at flower color. So he had one true breeding or purebred purple flower, and he had one true breeding or purebred white flower, and he'd cross the two with each other. That process right there is what we typically call hybridization. The offspring that you get from that cross are called hybrids. So the hybrids are going to be some combination of the two parent traits. The original parents that he started with was what he called the P generation, or the parental generation, and he would carefully control the matings of these two parents. So he would basically open up the flowers. He would remove the stamen, which is the male part of the flower, so that he was absolutely positive that the parent that he wanted to serve as the male was going to be the male. So he would go to another flower, collect pollen, and then basically using a paintbrush, you could do this with your finger as well, just take that pollen and place it on the female part of this purple flower, and now you know exactly which parent was the male, which parent was the female. Then he would simply wait for the seeds to develop. And once he had the seeds, those are the offspring right there. He would plant them out and then wait for them to flower so he could evaluate what colors they had. The first generation that he got was what he called the F1 generation. We want to be familiar with the P generation being the parents, the original parents, and that the F1 generation is the offspring that come from the P generation. Now, when he did this, he crossed the purple with the white, and he got the F1 generation, but he noticed that every single one of those individuals in the F1 generation had purple flowers. Not only did they have purple flowers, but they were just as purple as the original purple parent. If this was blending going on, we would expect that the F1s would be kind of a light purple in color. So right away, he did have results that did not coincide with the blending hypothesis. So he started to think perhaps there was something else going on here. In fact, it looked like the white parent did not contribute anything to these offspring. He then took two of the individuals in the F1, crossed them with each other, and he got what he then called the F2 generation. Notice that the F1 served as parents for the F2. When he looked at the offspring from the F2 generation, he noticed that he did get a lot of purple flowered plants, but he also did have some white colored flowers. This indicated to him that the white information was present in those purple looking F1 individuals, even though they weren't looking white. Not only did he have some purple and some white, but they were also present in a very characteristic ratio, and that was in a 3-to-1 ratio. So in this F2 generation, basically 3 parts were purple, or 75% was purple, and 1 part, or 25%, was white. So Mendel basically concluded that in the F1 plants, only the purple flower color was affecting the hybrids. And then he called the purple flower color dominant because it was what was being seen. And he called the white color recessive because although the information must be there, it was not really showing through in those particular individuals. Now, he didn't just look at flower color, he looked at a lot of different traits. He looked at flower color, flower position, seed color, seed shape, a whole list. In all of his experiments, he started with two true breeding parents that had contrasting looks to them. And in all cases, when he got the F1, only one of the two looks was apparent. That is what he called the dominant trait. So if we look down here at the seed color, when he crossed yellow with green, all of the F1 offspring were yellow. Then if he went on and took F1 offspring, crossed them with each other, then he would get the F2 generation. And over in this column, we see the numbers for his F2 generation. Notice in all cases that no matter what, the ratios did come out to be approximately 3-to-1. Now they're not exactly 3-to-1, but very close to 3-to-1. So just to sum up where he was at, he said that when I do these crosses, the F1 hybrids that I'm getting only express the dominant trait. So they only look like one of the two parents. Secondly, in the F2 plants, either the dominant or the recessive look can be present. Then he said, not only that, but in the F2, there is a specific ratio that I'm seeing. So the ratio of dominant plants to recessive plants is approximately 3:1. Mendel took this information, and this is where the law of segregation came from. So the law of segregation was just for the purpose, really, of explaining that 3:1 pattern that he kept seeing repeatedly. Keep in mind that Mendel was really working off of numbers here because he didn't know what DNA was, or he didn't know what chromosomes were, for that matter. So with the law of segregation, if we look at the basis of it, first of all, there are alternate versions of genes that account for variations in inherited characters. We call those versions of the gene alleles. In this case, we were looking at a flower color gene, and that flower color gene is going to have a specific locus or location on a chromosome. We had one allele that was purple and one allele that was white. For each character, or for each gene that we're looking at, an organism is going to inherit two alleles, one from each parent. Keep in mind that when we're talking about typical organisms, they are diploid. And because they are diploid, that does mean that they have two of each chromosome. Those are the homologous chromosomes. Well, as you see in this picture here, if we have a homologous pair of chromosomes, each one of those chromosomes is going to have a locus for the flower color gene. They may or may not have different versions of those genes on them, but they're going to have two alleles or two versions of the gene. What we see in this particular picture is what we had in the F1 individuals. Because the F1 individuals were always going to get the purple version from the purple parent. They were always going to get the white version from the white parent. The fact that the purple parent was always passing on purple and the white parent was always passing on white, that's really what's meant by the true breeding or purebred. Now, he went on to say that if they have two different versions, or if the two alleles at a locus differ, then the dominant allele will determine the organism's appearance, and the recessive allele will have no noticeable effect. So in those F1 individuals that were looking purple, it wasn't that the white wasn't there, but the white was not having a noticeable effect. So the purple color was somewhat dominating or covering up the white version. He also said that the two alleles for heritable character are going to separate or segregate during gamete formation and end up in different gametes. That right there is really meiosis that we're talking about. And if we're talking about the two alleles being on a pair of homologous chromosomes, the homologous chromosomes do separate or segregate during meiosis. So if we look at this in this slide here, we've got a simplified look at meiosis. We've got a diploid parent that has a B gene. There are two different versions of that B gene. We have capital B and lowercase b. Capital B is representing the dark blue, lowercase b is representing the light blue. In interphase, all of those chromosomes will be copied, so we'll get the replicated chromosomes. Then during meiosis 1, we get separation of the homologous chromosomes. So notice at that step, we are separating the capital B dark blue version from the lowercase b light blue version. And then ultimately, after meiosis 2, half of the gametes that are produced will have one version of the gene, half of the gametes will have the other version of the gene. That's the law of segregation right there. So notice that the capital B and the lowercase b, they are on separate chromosomes altogether. They do not mix together and create a new color, but one of the colors is capable of kind of covering up or masking the appearance of the second one. If we look at this in more detail with regards to Mendel's cross, if we look at a seed shape example, we've got a gene here for seed shape. There are two versions. We have round and then we also have wrinkle. Our parents are going to be diploid, and since they're diploid, they're going to have two alleles for this seed-shaped gene. we represent the alleles with letters. Typically, we represent the dominant version with the capital letter, and the lowercase letter represents the recessive version. Since we're starting with true breeding parents really means that they've got two copies of the exact same allele there. Notice that the round parent has two copies of big A, and the wrinkled parent has two copies of lowercase a. Now, if these are going to reproduce or cross with each other, the first thing that would have to take place would be that we would need to have meiosis occur. If meiosis occurs, we're going to get from the round parent all gametes, or all egg, all sperm that have one capital A inside of them. If we get meiosis with the wrinkled parent, all of the gametes are going to get the lowercase a. Since there's only one type of gamete from either parent, we have basically our egg here, and we have our sperm over here, and then it's just a matter of fusing the two together with the process of fertilization. When that happens, we get the F1 individuals, or the F1 offspring, and now they have one capital A and one lowercase a. Now, notice in the picture that that F1 individual looks round. It looks round because the capital A is covering up the presence, or the appearance really, of the lowercase a. But the lowercase a is still there inside that particular individual. If we go on to produce the F2 generation, to make the F2, we have to either self-fertilize the F1, cross it with itself, or take two F1s and cross them with each other. Again, if we're going to make a cross, the first thing that really has to occur is that we have to get meiosis take place. So if meiosis takes place, this is just like the example of meiosis we looked at before. Notice that if we look at meiosis, and we have two different versions there, we expect to ultimately get gametes that half of them have, in this slide, capital B, half of them have lowercase b. So if we go back over here, notice if we just change the letters to A, half of the gametes will have capital A, half of them will have lowercase a. We're going to take two parents that each have that combination. So the way that we look at all the possible combinations we can get is we're going to take the female's gametes and place them on one side of this box structure. So if they're female gametes, these would basically be our eggs. And then we're going to put the male gametes on the other side, and those would then be our sperm. We want to look at all possible combinations that we can get based on a random selection of eggs and a random selection of sperm. What we're looking at in this box is what we typically call a Punnett square. And in the Punnett square, this square was kind of come up with by a man with the last name of Punnett. We fill in the boxes by looking directly above and bringing the letter down and directly across. So in this first box, we would have two capital A's. In this box here, we would have a capital A and a lowercase a, and so on. Hopefully you get the idea as to how we would fill in the letters in the squares. What these squares are telling us is these are basically our potential F2 offspring. So we can see that we've got really 4 different combinations that we can get in our F2 offspring, and notice that quarter probability really of getting any one of those 4 boxes. Notice that if we throw in the looks, as long as there is one capital A there, they are going to have the round appearance to them. That is because the capital A is our dominant version. If they have only lowercase a's, then they're going to look wrinkled. So we can say that we've got these different combinations of alleles. A quarter of the offspring in the F2 we would expect to be big A, big A. Half of them we would expect to have one of each, and then a quarter of them to have little a, little a. If we look at the appearances, 3/4 of them, or 3 parts, will be round, and 1/4 of them, or 1 part, will be wrinkled. That's the 3-to-1 ratio that we were looking at before with our flower example. If we look at the flower example here, notice that we've got 2 true breeding parents, and by true breeding, they have 2 copies of the same letter. Now, this picture is from your textbook, but it does show you that you can pick any letter that you want to represent the gene. But I would advise you to steer clear of letters where the capital version and the lowercase version look very similar to each other, such as the P's. That often leads to just common errors when you're setting up your genetic crosses because you forget whether it was supposed to be capital or lowercase. If we look at it on here, though, when we have meiosis, this purple parent is always going to contribute a big P. This one, always a little p. When they combine, we get F1s that have one big and one little, and they look purple because the capital P covers up the lowercase. If we cross two F1s with each other, one parent will produce half of them with big P, half of them with lowercase p, and the other parent will produce half of them with big P, half of them with lowercase p. And then we fill in the boxes in this Punnett square just as we did before. Now, it does not matter whether you put the eggs on the top or on the side or vice versa. What is important is that the eggs are by themselves on one side and the sperm is by itself on the other side. Now, if we review some terminology, we want to understand that the hereditary determinant of a trait is a gene. So in these examples, we talked about a seed shape gene. We also talked about a flower color gene. It is very possible to have different forms of a particular gene, and those are called alleles. So round and wrinkled, those were alleles. The purple and white, they were alleles. When we talk about the combination of alleles that an organism has, that is known as the genotype. So the genotype is the genetic makeup of an organism or the cell. In the examples that we've seen, we could have two of the same letter there. We could also have two different letters present. So here's a look at some chromosomes, and they're showing you different genotypes that are possible. It is possible to have two of the exact same alleles, which is what we're seeing on this chromosome for the A. That would be what we call homozygous. So homozygous individuals have two copies of the exact same allele. So two capital A's, or it could be two lowercase a's for that matter. We could call them either homozygous dominant if they had two of the capital, or we could call them homozygous recessive if they had two of the lowercase version. Homozygous is another way to say true breeding. So in Mendel's examples, he was always starting with homozygous parents in the P generation. Now, it's also possible to have two different alleles present, like a capital A and a lowercase a, like we see right here. That is what we call heterozygous. Whether you write capital A, lowercase a, or lowercase a, capital A, that really is the exact same thing. However, the standard way to write this is to write the capital letter first. Now, when we use the terms homozygous and heterozygous, we're talking about one gene at a time. So in this picture, you see that we've got an A gene, a B gene, a C gene, D gene, and so on the whole way down. It is very possible to be homozygous for A, heterozygous for B, homozygous for C, and so on. So we don't have to be homozygous for every gene or heterozygous for every gene, and we are most definitely going to be some combination of the two, just depending on the particular gene that we're looking at. So we want to know what the genotype is, be able to use the terms heterozygous and homozygous. One final term that we want to be familiar with is what is called the phenotype. The phenotype is basically the observable properties of the organism. So what it looks like. So purple, white, that would have been the phenotype, round versus wrinkled. When Mendel was talking about that 3-to-1 ratio, he was really looking at a phenotypic ratio. That was a ratio of the phenotypes. If we go back and we look at the results of his flower color cross, notice here in the F2, because this is the F2 that we're looking at, in the F2, he had 3 parts purple and 1 part white. That was the phenotypic ratio. If we were to look at the genotypic ratio, he had basically 1 that was big P, 2 that were 1 of each or heterozygous, and 1 that was 2 little p's. That would be what we would call a genotypic ratio of 1 to 2 to 1. If you need to come up with a ratio, basically list the different genotypes that we have. Once you have listed them, then we simply go through and we count up how many we have of each one, and that will give you the ratio that we're looking at. Mendel's examples so far that we've looked at, these have been what we call monohybrid crosses, and that's because mono means one. So we're looking at one gene at a time. Now, since we are composed of thousands of genes, there certainly are more than one thing that's inherited at a time. So we will, as we get through this chapter, we will initially start with the monohybrid crosses. They are by far the simplest of all of them, and they are definitely the foundation for all of the larger crosses. So really important that you spend the time to really grasp what's going on in the monohybrid crosses. Now, notice in this slide that it is possible to have different genotypes here that all look exactly the same. So different genotypes can give the same phenotype. If Mendel was to go and just pick up a purple plant from the F2, he would not know if they were homozygous dominant, so 2 capital Ps, or whether they were heterozygous for that matter. If he did want to know what the actual genotype of that purple-looking plant was, that's where the test cross comes into play. So a test cross is a cross that takes an organism that looks dominant. It has the purple look to it in this example. And they try to figure out what the actual genotype is. So they take that dominant looking individual and they cross it with a recessive looking individual. The reason we cross it with something that looks recessive is because if it looks recessive, we know exactly, without a doubt, what the genotype is. There's only one combination that makes it look recessive. It would have to have the 2 lowercase letters. So if we cross this dominant looking flower with this recessive looking flower, we predict the outcomes. Notice that this parent right here, the little p's, are always going to segregate into little p and little p. So we know for sure what the top of our Punnett square is going to look like. It's going to have 2 little p's on the top of it. If we consider that this original one was big P, then we would expect that we would have 2 big P's down the side of the Punnett square. If that were the case, when we do our cross, we would expect all of our offspring 100% to come out looking purple. However, if this original one was heterozygous, we would expect this side of the Punnett square to have a capital P and a lowercase. And notice that we would expect half of the offspring that we get to turn out being white. So with a test cross, we make the cross and then we look at the offspring. If we get some that look recessive, so we get some white ones, then we can conclude, okay, this one must have been big P, little p. However, if we do the cross and we get lots of purple ones and no whites at all, then we can say, all right, we know for sure this was the homozygous dominant version in this one. So with the test cross, we do the cross, evaluate the offspring, and then that enables us to go back to that parent and determine what its genotype was. If we look at an example here, in tomatoes, the shape of the fruit is inherited, and both round and elongate fruit are possible. With this, we first want to come up with the genotypes of the 2 contrasting true breeding parents, and we're going to call this shape gene the R gene. So if we have 2 parents and they're contrasting and they're true breeding, what that's telling us is that the 2 parents are homozygous, because true breeding means homozygous, and they're contrasting, so we know that they have different versions of the R, meaning one parent is going to be 2 capital R's and one parent is going to be 2 lowercase r's. So that's the cross that we're making. If we conduct this cross and we now get F1 individuals, progeny is the same as saying offspring, and we're saying all of the F1 have round fruit. Okay, there's a number of things we can determine here. First of all, we want to know what the genotype of the F1 is. Well, we know that from this parent we are always getting a capital R, and we know from this parent we are always getting a lowercase r. So if we simply combine those 2 together, we are going to expect all of our F1 offspring or progeny to be big R, little r. We then ask which trait or allele is dominant. We know that the capital R is dominant. We always use the capital R to represent the dominant version, but we know that that goes with the round look because all of the F1s look round. Which trait will be recessive will be just the opposite. It's going to be the elongate ones, and that would be represented by the lowercase r. Now, we want to know how many gametes can each of the F1s produce. So if we're going to cross them with each other, keep in mind that this one, half of the time, is going to give capital R. So we'd expect that to be half, and we'd expect this one also to give half lowercase r. So that's really what we're looking at there. If we take two parents that are doing that and we cross them together, that would be how we get the F2. So if we produce the F2, we should be able to figure out what proportion or percent of the F2 we would expect to be round, and then we can also figure out what proportion would be elongate. To do that, we simply take one parent's gametes and put it across the top of our Punnett square. We take the other parents and put it down the other side, and we're crossing two F1s, which are both going to have a capital R and a lowercase r. Then we simply fill in the boxes. And notice that in this case, this box, and this box all have one capital R. So we would expect that 3/4 or 75% of our offspring would be round. And then the flip side of that, only this one last little box here is going to be elongate. So we'd expect 1/4 or 25% of them to be elongated. That's a run through of one example of a monohybrid cross. We want to make sure we understand that true breeding means homozygous, and it is possible to be homozygous dominant, which this would be homozygous dominant, or homozygous recessive. Here we've got F1s that are heterozygous. We want to make sure that the dominant allele is represented by the capital letter, the lowercase allele represents the recessive version, and that we understand when we are making the F2 that we use two F1s as parents. So that was what we were crossing with each other. If we look at yet another example, if we look at a human trait, widow's peak is a dominant trait in humans. This is a little peak point that comes down in the hairline on the forehead. I want to say that consider we have a heterozygous woman. So if she is heterozygous, she has two different versions of the gene. She has a widow's peak, and she marries a man who does not have a widow's peak. So we want to use the letter A to represent this gene, and there's a number of questions we're asking about them. First off, we want to know what is the phenotype of the woman. Keep in mind that the phenotype is the look. Okay, so here we're talking about widow's peak. Do they have one? Do they not have one? So as far as her phenotype, it's just that she has a widow's peak. Her genotype, we're given a clue up here that she is heterozygous. So when we write that, we want to write the capital A first and the lowercase second because that is the standard way that it's written. She's got one of each. Because widow's peak, having the widow's peak is dominant, that big A that she does have causes her to have that appearance. As far as the man is concerned, notice that he does not have a widow's peak, so that would be his phenotype. And then his genotype, the only way that he cannot have a widow's peak, which is the recessive look, would be if he happened to have two copies of the recessive version. Because if he had at least one capital letter there, he would have a widow's peak. Once you figure out the genotypes of the parents, the rest of the question is really just doing the cross. So we want to know what is the probability that these two individuals will produce children that have a widow's peak. Well, the cross that we're making is we're taking the woman, we're crossing her with the man. First thing we would need to do is we need to figure out what are the gametes this parent can produce. She can contribute either a big A or a little a. This one, either a little a or a little a. So same thing there. Then it's just a matter of filling in the Punnett square. One parent's gametes down the side, other parents across the top. If we fill in the boxes, here are the combinations that we get. So the probability of getting children that have a widow's peak, notice these top two boxes do have a widow's peak. So this is going to be 50%, or you could also say 1/2.